and I'm really excited to hear what you all say. I'm really looking forward to it. Should be awesome.
Thank you, Colin.
Colin, we are ready to go whenever you are.
Terrific, thank you. All right, hi everyone again. And this afternoon's session, we have the researchers that were given awards in 2018. This all started with fundraising in 2017. And we went through a very lengthy process selecting the awardees. At first we thought we'd only have enough money for one award. We had enough money for 2 awards, and then we had enough money for 3 awards. So it's getting towards the end of the 2 years that those projects are all running for. I think they might have been even closer to finished by now if it hadn't been for the COVID-19 this year. But we're all very excited to hear from Dr. Nemenoff, Dr. Awad, and Dr. Gainor about the projects. First, we're going to hear from Dr. Upal Basu. Dr. Basu is the Director of Scientific Research at Lungevity, and Dr. Basu is just absolutely incredible. He is emails, texts, phone calls constantly with us and with the doctors, and he's just a remarkable person. And he's going to be giving a bit of an overview about how the selection process works. He'll be speaking for about 5 minutes on that. And then Dr. Awad and his colleague, Dr. Chiarle, will be speaking about their project. And then Dr. Gainor and Dr. Jess Lynn from MGH will be speaking about Dr. Gainor's project. And then finally, we'll have Dr. Nemenoff speaking about his project at University of Colorado. And there'll be plenty of time at the end for questions and answers. So just type your questions in on the chat, and at the end we'll be able to answer some of those questions. Questions that can't get answered, we'll try to get answered later on. So don't be shy about asking your questions. So with that, I'll pass you on to Dr. Basu and get the ball rolling.
Wonderful. Colin, thank you so much for a very, very generous introduction. You're very kind. My name is Dr. Upal Basu, and I'm the Vice President of Research at Lungevity Foundation, and have been incredibly privileged and honored to work alongside a group of amazing members from the ALK-positive community and a group of phenomenal researchers from whom you'll be hearing from today. And again, thank you very much for a great partnership, and looking forward to doing great things with all of you. And I'm going to quickly share my screen and do a very quick overview of our research project and essentially how we've sort of selected grants. So as I just mentioned, my name is Dr. Upal Basu. And before I actually sort of get into the presentation per se, oops, I think I'm having a little bit of tech issues, I apologize. Before I actually get into the presentation, I actually wanted to start my presentation with this beautiful picture, and I call this my Lung Cancer Love Fest and Unity picture. And this picture was taken earlier on this year. And as you can see, there are a lot of familiar faces, and this is actually the last time I got to see this amazing group of people, because after that, the COVID-19 pandemic hit, and then everything went virtual. So all this is to say that, yes, I'm sitting in my apartment in New York, and I wish I was there to give all of you hugs, but sending you a lot of love and virtual hugs your way. Now, moving into the process that Lungevity Foundation used in partnership with the ALK-positives to fund research. As Colin mentioned, this was the first ever scientific project funded by the ALK-positives in partnership with Lungevity Foundation. So you guys took the leap of faith with us in 2017, and that was great. And in 2018, we actually announced the awardees. And as Colin mentioned, the initial goal of funding research was to actually fund one project. But thanks to you guys and all your fundraising efforts and all your passion to make a change in ALK-positive lung cancer, we were able to fund 3 projects, and you'll be hearing from the 3 awardees today. Now, how did we end up how did we end up with 3 awardees? What did the research process look like? So in a nutshell, Lungevity Foundation uses the NIH study section process. Now, the NIH is National Institute of Health. They are the largest public funder of research in the United States, and they follow a specific mechanism to select best research projects. So what did we do in partnership with the ALK-positives? We issued something called a request for applications, which is also known as an RFA, in November 2017 during the Lung Cancer Awareness Month. And we received 14 letters of intent, or LOIs, in January 2018. Now, these 14 LOIs were very, very specific to ALK-positive lung cancer. These 14 LOIs were then reviewed by a panel of experts, and we call that panel of experts a study section group, and I've defined what a study section is below. Essentially, a study section is a group of ALK-positive survivors who some of you know as the research review panel. We have a team of ALK scientific leaders, some of who are already on the phone and on the call today. And then we had biostatisticians who were charged with reviewing the applications. So we received 14 LOIs, and then based on the reviews we received from the panel of experts, we brought that down to 7 LOIs, and these very promising LOIs were then invited to submit a full application. Now, what is an LOI? An LOI is basically a sneak preview of what the entire proposal is going to look like. And why did we have this 2-step process? First, we had LOIs, and then we had full applications. That is actually to make sure that we really fund the best projects. So we had 7 full applications that we received in 2018, in April 2018, and then we had a study section where this group of experts got together on a phone call together, and then they decided what they wanted to fund. And then we announced the research awards in June 2018. And what does this really look like behind the scenes? And I wanted to give you a very sneak preview of what this really looks like. So on the left top corner, you see 5 members of the research review panel. These are actually members of the ALK-positive community. They're all incredibly attuned to the science of ALK-positive lung cancer, and they deeply care about making sure that the best research projects are funded. In the bottom left side, in the bottom left corner, what you see is the panel of ALK-positive experts who we brought together along with the research review panel to review applications. And then in the center, you see the Lungevity team. So these 3 groups of people were intimately involved in this process. And what did that really look like? So the research review panel and the experts reviewed 166 pages of LOIs. They reviewed 350 pages of grants, and the whole team spent about 975 number of hours on reviewing grants and progress reports. And that number is actually incredibly high. And you might be wondering why I'm saying 975 hours, right? These are actually volunteer hours. You have to understand that the research review panel, this panel of experts, they volunteer their time to review these applications to make sure that the best applications are funded. And all this is to say that you have this incredible team of experts backing your applications, this incredible team of experts making sure the best projects are funded. Now, you will be hearing from our 3 awardees very shortly, and I'm not going to steal their thunder, but you'll see a very common theme, which is sort of all pervasive. All 3 of the researchers we'll be hearing from, they'll be talking about how we can turn on the immune system to fight ALK-positive lung cancer. And then we just this year sent out this 2nd RFA for ALK-positive lung cancer. And again, thank you very much for your partnership, for taking this leap of faith with us again. And this is the biggest patient-driven lung cancer RFA to date. This is incredibly exciting. And again, to give you a sneak preview, so we finished reviewing the LOIs, we finished reviewing the full applications, and now the board of directors of the ALK-positive team, they're in the process of finalizing what the award, who the winners will be. And just to give you a very sneak preview of what the study section looked like, this was actually done on Zoom, and you can see all these amazing people getting together, discussing all these grants, and making sure, again, the best research projects are funded. And with that, I will end my introduction, but I would be remiss, being a public health scientist as well, I would be remiss in not leaving you with this message. Please continue to wear a mask. continue to social distance and continue to wash your hands because of the COVID-19 pandemic. And with that, I will hand it back to our moderator, Dr. Barton.
Thank you for that, Dr. Basu Roy, and we'll now move on and have the first of our presentations. So I'm excited to hear this.
Great. Well, hello everyone. Can you all see my screen? Yes. Well, I'd like to extend a warm welcome from Boston to everyone out there, and I wanted to extend a huge thanks to the Lungevity Foundation and the ALK-positive community for the opportunity to do the research that we'll be discussing. My name is Mark Awad. I'm the clinical director for our thoracic oncology group at the Dana-Farber Cancer Institute here in Boston, and I'm presenting today in close collaboration with Roberto Chiarle, who is professor of medicine, professor of pathology at Harvard Medical School at Boston Children's Hospital. So we'll have about 10 minutes to give you a very brief summary of what we've been doing, as well as give you a sense of where we'd like to go in the coming years with this work. So we know that there are many different therapies available for lung cancer. Among patients with ALK-positive lung cancer, of course, we have a number of wonderful ALK kinase inhibitors, some of which are shown here. And these therapies can work very well, they can work very rapidly, but one of the struggles for us as a community is the development of acquired resistance to these therapies. By contrast, in lung cancers that do not have targetable mutations, we tend to treat these cancers with immunotherapy alone, with or without combining this with chemotherapy. And here, although a minority of patients develop durable responses, there is this great potential for very durable long-term immunologic responses when using immunotherapy for these types of cancers. And just very briefly, how do these immunotherapies work? Well, on this schematic here, you can see a diagram. On the left is a lung cancer cell, on the right is a T cell of the immune system. We know that some immune cells can recognize lung cancer cells on their own and try to attack these cancer cells. But some lung cancers can escape from the immune system by putting up these stop signals, one of which is shown here called PDL1. And we know now that we can intervene and try to take down these stop signals with so-called PD1 or PDL1 inhibitors. These are drugs such as pembrolizumab or nivolumab, also called Keytruda and Opdivo. And these drugs work by essentially blocking the stop signal to re-engage the immune system to attacking lung cancer cells. And these therapies can work extremely well. Here's one of my patients at Dana-Farber, who is a 68-year-old man with KRAS mutant lung adenocarcinoma, diagnosed a few years ago now with cancer both in the left and right lung, as well as in other sites of the body. And you can see that this patient has been on pembrolizumab immunotherapy for over 3 years now and has had an amazing, wonderful, and durable response to these therapies. And the question that many of us have been asking is, can we stimulate the immune system specifically to attack ALK-positive lung cancer? And through work from many of my colleagues, we know that the currently approved immunotherapies that I was just discussing, such as pembrolizumab or nivolumab, are rarely effective against ALK-positive lung cancer. We do know that ALK as a protein is an attractive target for immune targeting. The protein itself is typically expressed only in cancer cells and is not in normal cells. Therefore, we hope that by attacking the ALK protein specifically, that will spare some side effects to normal cells. We know that ALK-positive cancer cells are exquisitely dependent on ALK for their survival. And we know that the ALK protein itself can be recognized by the immune system through some of the data that we wanted to share with you. So with that, I'd like to turn over some of the discussion to my colleague, Roberto Chiarle. And Roberto, you can please introduce yourself, and I'll advance the slides for you for the next few slides.
Yeah, thank you, Mark. So yes, my name is Roberto Chiarle. I'm originally from Italy, and I moved to Boston in 2012. And one of the reasons for this move was exactly to try to move one of our ideas, which is the ALK vaccine, to the clinical arena. And I was lucky enough to meet Mark early at the time. It was now a few years that we are collaborating, and we basically started with this work in which we demonstrated that a subset of ALK-positive patients carrying either lymphoma or lung cancer in this particular case, they can develop spontaneous immune responses against the ALK as a protein. So they see the ALK, which is the oncogenic driver of the tumor, as an antigen. So the immune system recognizes that. And patient can develop antibody and can also develop T-cell responses. And so based on that, we, and Mark, you can advance the slide, please. We are basically facing a sort of conundrum here, because we know that ALK is a tumor antigen. So why immunotherapy isn't effective in ALK-positive non-small cell lung cancer patients? So that's a kind of conundrum that we are trying to solve now. And more importantly, how can we make an unresponsive ALK-positive lung cancer treatable with immunotherapy? And in this case, and Mark, you can advance, we developed the concept of generating an ALK vaccine that could be the solution, because we think that the problem is in the threshold and intensity of the recognition by the immune system of the specific ALK antigen. So we started, of course, working in mice to prove that this hypothesis was true. And in a paper that we published a few years ago, we clearly demonstrate that a formulation of a vaccine was able to induce a very strong immune infiltrate in tumors. So you see here on the right, basically, a lot of immune cells that infiltrate the tumor, and of course the tumor gets smaller and basically is eliminated by the tumor cells in mice. And on the right side, you can see how many more T cells, which are the effector arms of the immune system, we can bring inside the tumor after vaccination. And in the next slide, basically we proved that this approach in vaccination is able to extensively and for a long time, here is up to 20 weeks, which is a long time in the mouse lifespan, can basically block the tumor growth in a significant manner. And what is more importantly, and is the most recent data that is still unpublished, and Mark, you can show the next part of the slide. Exactly. So this is probably the most important piece of data that is really giving us a lot of enthusiasm to bring this concept to the clinical arena in a phase 1 clinical trial. So if you see here by the color line, in black is a tumor, an ALK-positive lung tumor in mice that is left untreated. So you see the survival is very short. Of course, if you don't do nothing, mice, they die quite rapidly. If you give the mice an ALK inhibitor, in this case, we are using lorlatinib, which is a 3rd generation ALK inhibitor, you can extend survival pretty nicely. Here is the blue line. And if you add a little bit of immune checkpoint therapy like anti-PD-1, still you don't make a big difference. Actually, the two curves look exactly the same. But what is striking is in any condition in which you add a vaccination, the red curve, the purple curve, and more impressively, the green curve, which is a combination of vaccination, an ALK inhibitor, and immune checkpoint therapy, now we can completely cure mice, a large fraction of them, up to 80% of them. And these mice are completely free of tumors. They are basically now healthy mice, and the survival is extremely long, almost up to 1 year, which is basically equivalent to more than 15 to 20 years in a human lifespan. So we were very, very excited about this result, which finally we can cure the tumors completely by a combination of an ALK inhibitor, a vaccination that decreased the immune infiltrate, and eventually immune checkpoint therapy. And so I think in the next slide, I'm returning the handle to Mark, which will introduce how we think to bring this concept to the clinical arena for the phase 1 clinical trial. Mark.
Thank you so much, Roberto. So Roberto's done an amazing work developing some very powerful tools in the laboratory of ALK-positive lung cancer in mice. And through doing so, we're now hoping to extend these findings into first in human phase 1 clinical trial. And we've been collecting many samples from patients with ALK-positive lung cancer over these past several years, and we've identified several peptides, which means short pieces of the ALK protein, in which we think there can be a particularly potent immune response against these ALK peptides. And in addition, we're collecting samples from many more patients to try to identify even more ALK peptides that we could potentially use for therapeutic purposes. To start, we've proposed a relatively small trial of 24 patients. In the first cohort of 12 patients, those who are developing disease progression on an ALK inhibitor, such as alectinib or lorlatinib, could stay on that ALK inhibitor, and then we would add an ALK vaccine on top of their oral ALK kinase inhibitor. In a second cohort, among patients who have had prior ALK inhibitors and or chemotherapy but have not yet had immunotherapy, our plan is to combine immunotherapy with a PD-1 inhibitor together with an ALK vaccine. This is a general schematic of the schedule for patients in whom they would receive the vaccine at regular intervals, initially more frequently, followed by these booster shots, along with immunotherapy in the second cohort. Our hope is to also do biopsies at baseline as well as after vaccination to see if we can replicate what we found in the laboratory to see, does the immune system really then infiltrate and go into these tumors after vaccination? We've currently raised $750,000 through a generous grant from the Koch Institute for Integrative Cancer Research, which should fund about 50 to 60% of the cost for this trial. And we've partnered with a pharmaceutical company named Elysia Therapeutics to cover the cost of vaccine synthesis. So we're very excited. We're hoping for the final bit of fundraising to be able to fund this trial, which typically costs about $1.2 to $1.5 million for a trial of this. So to conclude, we know that a greater understanding of the interplay between the immune system and human cancer is really vital and critical to the development of novel treatments, and a therapeutic ALK vaccine will hopefully help patients with ALK-positive cancers live long and live well. We really wanted to thank, again, the Lungevity Foundation and the ALK-positive community, without which this research would not have been possible. And through the generous and tireless fundraising through these foundations, it's really fueled our understanding and our drive and excitement to develop these laboratory findings and bring it into the clinic for a human trial. So with that, I'll stop and we'll have some time to discuss at the end.
So thank you so much, Dr. Awad and Dr. Chiarle. And I know that there's so much interest in the group about your projects, and everyone's rooting for you 110% to move it along quickly and most importantly, successfully. And we'll now move on to hearing the presentation from Dr. Gainor and Dr. Len.
Great. Thanks so much, Dr. Barton. And it was a great presentation by Dr. Awad and Dr. Chiarle. And you know, I think in many ways, as you'll see, all three of these projects really complement one another. And I think really viewing it as not just a collaboration between researchers in the ALK-positive community, but also collaborations among these researchers. So my name is Dr. Justin Gainor. I'm the Director of Thoracic Oncology at Massachusetts General Hospital. And, you know, I like Dr. Awad, also want to extend a special thank you to Lungevity as well as the ALK-positive community. For me, this is a really special award because it comes from you, the patients. And as a result, it's incredibly inspiring and motivating, you know, to do work. And I think it really, you know, drives us to try to get insights and bring them to the clinic as fast as humanly possible. And so on this particular project, you know, I'm so grateful that I've been able to partner with Dr. Jessica Lin. Dr. Lin is really a superstar clinical translational researcher. She's currently one of the leading investigators for the ALK Master Protocol. And, you know, she's someone who has already been able to translate findings from the basic science laboratory into the clinic. And so I'm really fortunate to work with her on this. And so Dr. Lin, I'm going to hand it over to her, let her introduce herself a bit more, as well as give you an overview of where we are on this project.
Thank you, Dr. Gainor, for such a kind introduction. Hello, everyone. I'm Dr. Jessica Lin. I'm a thoracic oncologist at the Massachusetts General Hospital Cancer Center. And my focus is really on taking care of patients with ALK and ROS1 rearranged lung cancer and developing new therapies. Dr. Gainor has been so generous to offer me this platform to connect with many of you, but please rest assured that both he and I will be available during the Q&A session to address many of your questions. We are so excited to be here. We wish that we could be meeting with all of you in person, but I really want to congratulate all of the organizers. It's been such an exciting meeting already virtually, and we want to send all of you our virtual hugs. Thank you again to Lungevity and ALK-positive, and also to Dr. Gregory Riley at Memorial Sloan Kettering, who's really a key collaborator for this project as well. And so taking a step back, you've heard already today earlier in the morning about how our treatment of ALK-positive lung cancer has been transformed by these targeted therapies with ALK inhibitors. And so ALK rearrangements, as you know, were first discovered in 2007, but over the past decade, we've seen really a very rapid development of multiple ALK inhibitors that have now been FDA approved, which includes crizotinib, which is what we call the first generation ALK inhibitor, and then the second generation ALK inhibitors, ceritinib, alectinib, and brigatinib. And then in 2018, the third generation inhibitor, lorlatinib, which Dr. Teddy Johnson spoke to us about in detail earlier today. Here in the US, we are fortunate to be able to treat our patients with one ALK inhibitor after another in a sequential manner. So as many of you know, during this time, if you're newly diagnosed with advanced ALK-positive lung cancer, we'll commonly reach for alectinib as the first ALK inhibitor. And then if there is disease progression on alectinib, we'll discuss several options, but these include lorlatinib or perhaps another second generation ALK inhibitor like brigatinib. Of course, the challenge and the need for additional treatment comes from the fact that there is often acquired resistance to these ALK inhibitors that we face in patients in clinic. And so these pie charts are quite complicated, but I wanted to use them to highlight one point today. These pie charts are looking at biopsies that were taken from our patients with ALK lung cancer who were experiencing disease progression on a second generation inhibitor like alectinib, for example, or brigatinib, or progression on lorlatinib. And these pie charts are looking at different new ALK resistance mutations that are found from sequencing of those biopsies. And so after alectinib or brigatinib, in many cases, we detect these new ALK mutations, often just a single mutation. Whereas after lorlatinib, what we find is that actually many of the patients, if they do have ALK mutations that are driving that resistance, are occurring in double or even triple the number. So there are 2 or 3 new different mutations that we can detect in the ALK gene. And then there are also many patients where we don't detect new ALK mutations, where the hypothesis is that tumor cells have figured out another way, bypassing the ALK pathway that allows them to grow despite therapy. And both of these scenarios really pose a challenge for us in thinking about how best to overcome this resistance. And that's where immunotherapy and the hope for immunotherapy comes in. And so you've already heard from Dr. Awad about the concept of the immunotherapy. This was actually one of the major breakthroughs, not only in lung cancer, but in many different tumor types. And the idea of immunotherapy is to really unleash your body's own immune response against cancer and its ability to fight off cancer. And so the PD-1, PD-L1 inhibitors that Dr. Awad mentioned, which we also refer to as checkpoint inhibitors, are these IV infusion drugs that are one type of cancer immunotherapy. And they've shown good success in a subset of lung cancer patients. And in fact, some of the patients, when they do respond to these checkpoint inhibitors, they can have these really long, durable responses too, which is part of what generated a lot of excitement and enthusiasm. But the fact of the matter is that there are many patients who also don't benefit from these checkpoint inhibitors that are FDA approved. And what we've learned over the recent few years is that our patients with, for example, EGFR mutant lung cancer, you heard about from Dr. Kamich, or ALK-positive lung cancer, don't seem to derive as much benefit at this time from these checkpoint inhibitors when they're used in clinic. So there have been a number of studies looking at this. These graphs are from actually one of the earlier studies that were performed by Dr. Gainor and published back in 2016. And what he did here was to look at the responses to checkpoint inhibitors among patients with EGFR mutant or ALK-positive lung cancer and compared that to patients without EGFR mutations or ALK rearrangements in their lung cancer. And so the graph on the left, what that is showing is that if you have EGFR mutant or ALK-positive lung cancer, the rate of response to these checkpoint inhibitors that are currently available to be prescribed, like pembrolizumab, nivolumab, for example, are quite low, 3.6%, compared to over 20% in those who don't have EGFR mutations or ALK rearrangements in their tumor. On the right, you're looking at a curve showing progression-free survival. So Dr. Kamich explained what progression-free survival is. This is kind of the length of that time between initiation of treatment, in this case, the checkpoint inhibitor, and the time that the cancer starts getting worse or you're getting worse. And so what you want is a longer progression-free survival. And here, what you can see is that patients with EGFR mutant or ALK-positive lung cancer on these checkpoint inhibitors have shorter progression-free survival. And what we want to do is do better than this. So that leads us to our key question of the project is, how can we really try and extend the transformative benefit that we've seen with these immune therapies in some patients with lung cancer and bring that to our ALK-positive lung cancer patients? So to develop and to inform the development of these distinct immune-based strategies that may be required in treating ALK-positive lung cancer, Dr. Gainor and the team have proposed a really systematic and multi-pronged approach to examine factors not only in cancer cells, but also in cells like immune cells and inflammatory cells that are surrounding the cancer cells that may be important in regulating immune responses in ALK-positive lung cancer. Now, one of the challenges there, you might imagine, is that while ALK-positive lung cancers affect many patients, as all of you in the audience today know, really, if you take a step back, it still remains a relatively small subset of non-small cell lung cancer as a whole. So ALK rearrangements occur in anywhere between 3 to 5% of non-small cell lung cancer. And so when we're trying to look at genome and immune landscape of ALK-positive lung cancer in detail and really at a large scale, that becomes a challenge. And so the strategy that we've come up with is to partner with the ALK-positive community to establish an infrastructure that facilitates the acquisition of tumor specimens and clinical data at a larger scale that is not geographically limited. For example, we don't have to just look at patients that are able to come to Boston or New York, that we can enroll patients from across the nation in trying to perform this study. And so we've been collaborating with the Adario Lung Cancer Medical Institute, abbreviated ALCMI, in order to design a web-based, patient-driven platform that allows us to perform this study. So what that allows us is there's a web-based portal, for example, that patients can go on and provide an IRB-compliant electronic consent, informed consent, that allows us to then acquire clinical data and tumor specimens that can allow for immunologic studies. So this study is already fully written up. It is entitled Enigma Plus, So Enigma Plus is short for elucidating novel immune and genomic markers for ALK-positive lung cancer. And we're excited to let you know that the study is near IRB approval, and we hope to be able to launch this study and have patients participate within the next couple months or so. And so we wanted to really take this opportunity today to let you all know about our Enigma Plus study. And for those of you who are interested, we'll definitely make sure to let you all know about the website that you can go on to learn more information. So just to really quickly give you an overview of what information then we can try and gather once we have that clinical data and tumor specimens. As one example, what we'll try to do is define what we call tumor intrinsic factors that can be associated with immunity in ALK-positive lung cancer. So an example of that is looking at something called tumor mutational burden. And what that is the overall kind of amount of mutations across the different genes that are present in your specific tumor. The reason we're interested in that is because there are studies that have shown us that the more mutations there are in general, the higher the likelihood that there may be new novel proteins, what we call neoantigens, that may be important in stimulating and triggering immune response. And so we have some preliminary data shown here that suggest that in ALK-positive lung cancers, this tumor mutational burden may be lower than what we see in non-ALK-positive tumors, but we are really hoping to look at this in a much larger scale across the population. As another example, we want to look at this marker PD-L1 that Dr. Awad mentioned earlier, a marker that we think is associated with immune checkpoint inhibitor responses, but perhaps not in all patients. And also look at other checkpoints other than PD-L1 and immune cell populations that may be relevant in thinking about galvanizing immune responses in ALK-positive tumors. And then we're also looking at what we call anti-ALK T-cell responses. So actually there are many different types of immune therapies beyond checkpoint inhibitors. So one example is what we call cellular therapy, where what you do is try and harvest the patient's T-cells and modify them and alter them in some way that augments the ability to mount an immune response against ALK-positive lung cancer. And that is something that we're looking at enabling for ALK-positive lung cancer, but there are many steps that have to be accomplished before that becomes a reality. And so we are looking at blood samples from ALK-positive patients to see if we can isolate reactive T cells against ALK. Lastly, at the end of the day, once we've learned important lessons about immunology of ALK-positive lung cancer, we hope that we'll find a venue by which we can share all of the important lessons with the ALK-positive community and the broader research community. Some of you may have seen the CBio portal, which is a web-based portal that was developed by the Memorial Sloan Kettering Cancer Center. And it allows easy visualization of large-scale aggregate data, looking at the clinical outcomes, the mutation count, protein expression data, and it's available to the public.
Can we have enough time to let Dr. Nemenoff do a presentation also? And
sorry about this. So we are hoping to do something.
We'd love to hear you talking all day.
Yeah. And so I'll just finish by acknowledging patients and families, all of you. And also we want to acknowledge Dr. Ali Shah, who's really been pivotal in the development of many of the available ALK inhibitors while she was at Mass General. And then other ALK research members at our institution, including Dr. Dagago Jack, Dr. Hara Yoda, and Jamie Heather. And thank you very much for your time.
Thank you so much, Dr. Lin. Just to everybody, please give a round of applause, even though they can't hear you. All these speakers have given up time on their Saturday and spent, as you can tell, lots of time preparing before today, and we're extremely grateful. So thank you so much to all the speakers. Dr. Nemenoff, you are on.
Thank you. I just want to echo my great appreciation for Lungevity, but in particular the ALK positives and the support of the patients and actually being able to interact with the patients. For me, I'm a basic scientist, and you tend to get cooped up in the lab a bit. And for me, it is really always an eye-opening and a wonderful experience to interact with the patients. And in fact, the research that we have undertaken would never have happened without the support of the patients. So I really want to basically thank everyone, and also thank you for inviting me to present on this project. So our project is entitled Complement Inhibitors as a Novel Therapeutic in ALK-positive Lung Cancer. And again, I think I'm not going to go through a lot of detail because you've heard terrific talks from the two previous speakers, the two previous groups. But again, I think I wanted to sort of stress the fact that immune checkpoint inhibitors, which have gotten a huge amount of press and have really changed the way I think a lot of us think about cancer treatments, but these have really low efficacy in ALK-positive lung cancer, as you've heard from the previous speakers. However, that doesn't really say that the immune system is not participating. And you've heard some really nice approaches from the two other groups, and there are other people working on this to try to develop a much better understanding of how the immune system and the tumor microenvironment in general affect the progression of ALK-positive lung cancers. And it may not be immune checkpoints as monotherapies. These are unlikely to work, but I think the goal of a lot of people's work, including ours, is to really understand in ALK-positive lung cancer, how does the immune system participate, how can we rev it up, and how can we develop new therapeutic approaches that target this. And the hope would be that these would be used in conjunction with targeted ALK inhibitors. And if they're effective, they really, as you've heard again, really prolong the duration of response. So we're interested for quite a long time in the complement pathway. And one of the things that is always a little off-putting about our presentations is this is an incredibly complex system, but I'm going to just sort of highlight the importance of it. So this is a very ancient part of the immune response. It's part of the innate immune response and involves these complex series of deposition of proteins on cell surfaces, but it's a way that your body recognizes infection very rapidly. But importantly, it also communicates with the adaptive immune system. So we've been interested in this for quite a while in the setting of cancer. And there's been work a long time ago. It was thought that perhaps the complement system was irrelevant, or perhaps it enhanced responses. But it's become clear for at least the last, I would say, 5 to 10 years that in many cancers, if you activate complement, which occurs frequently, it actually is a bad thing, and it leads to cancer progression and metastasis. And without going through all the details of this whole system involves the deposition of a protein called C3, but importantly, there's the production of these fragments of these proteins, which are inflammatory mediators. And so the way that it's thought that complement actually promotes cancer is that this activation results in these inflammatory mediators. You bring in inflammatory cells, you create this chronic inflammatory state, which is associated with cancer progression. And it's clear that this also impacts the ability of the adaptive immune system to block this. And so it's been shown by our group and a lot of other groups that in fact complement activation is one of the ways that the adaptive immune system, CD8 T cells and CD4 T cells, which you heard about in the last talk, the way they're shut down. So it's an alternative way to block immune attack of cancer cells, similar to what checkpoints do, but this is a way that the innate complement system works. So we've been interested to try to understand, is this active in ALK-positive cancers, and can we harness this to develop new therapeutic strategies? So the two aims of our project, which was funded in 2018, were to define mechanisms whereby complement activation mediates progression, and then more translationally and importantly, test a panel of complement inhibitors as therapies for ALK-positive cancers. So basically, we've used a mouse model, which I'm not going to go into a lot of detail, but these are two ALK-positive implantable cancer cell lines. So we implant these into mice, we let them establish tumors, and then we treat them with these complement inhibitors. And so like you can see here in both of these, is if we use a genetic approach, so we can knock out complement in a genetic way, but we can use these small molecule inhibitors of these two parts that I talked about, the C3a fragment and the C5a fragment. And both of these show great efficacy and, in fact, can eliminate tumors almost completely in these two models. So this is preclinical published data suggesting that complement activation could be an important way to treat these cells. We've also shown, I don't have time to go into this, that all of these effects of complement are mediated through a very complex changing of the whole nature of the tumor. So in this experiment, we're actually using a technique called molecular cytometry, which allows us to profile all of the changes in these tumors. And it says, so what happens if you give this inhibitor? You're shrinking the tumor, but what other effects are you having? And I'm not going to go through all of these. Each of these clusters represents a particular population of cells that are in the tumor. So these could be vascular cells, they could be inflammatory cells. And, you know, very simply what you can see is that some of these clusters appear when you treat these cells with complement inhibitors. So these are a control tumor we've analyzed this way and 2 tumors treated with complement inhibitors. So all this says is complement is effective, but it also has marked changes in the tumor microenvironment. In the last year, we've begun to look at this in patient samples, and we've done some multispectral imaging of ALK-positive tumors, and so far we've looked at about 7 of these, and we can identify complement activation in a significant portion. We've made sort of an arbitrary cutoff here, but of these initial 7 tumors we've looked at least 3 of them seem to have a pretty robust complement activation. So from a perspective of treatment, it would seem appealing that if this pathway is activated in a large number of ALK tumors, it could be targetable. So what I've shown you from so far is that complement activation leads to progression, inhibitors block progression, and it's frequently observed in human ALK-positive tumors. So where does the landscape stand in terms of inhibitors? Since this pathway is complicated, there are many possible ways to target this. What attracted us from the very beginning was the fact that eculizumab, which is a drug that blocks complement C5a production, is actually an FDA-approved drug for a number of kidney diseases, which I know very little about, such as paroxysmal nocturnal hemoglobinuria and atypical hemolytic uremia.
Dr. Nemenoff?
Yeah.
I think we're pretty much out of time, unfortunately.
Can I show one last slide?
Yep.
Okay, so one last slide. It has been difficult, and I'll wrap this up in less than a minute. It has been difficult to move some of these drug companies, but I wanted to we're really excited by the fact that there is now an active multi-institutional trial involving a complement inhibitor on all solid tumors, which has just been opening in our institution. So I think the lag in moving these drugs into the clinic is being overcome. And I wanted to really just reiterate my enthusiasm that these drugs, which are safe in patients, could be effective in ALK-positive tumors. And I'll end there.
So we're not going to have any time for answering the questions, but certainly your questions can get answered later on. To all the speakers, I know that you've excited our members, and I can tell you with the latest round of proposals that we're reviewing at the moment, there's even more excitement. So as I keep saying to myself, just stay long enough and the research will come along to keep us alive even longer, and then stay alive a bit longer, and the next lot of research will come along to keep us alive even longer. So as I always say to the members in our group, go research. And thank you so much to all of you.
Thank you.
Hello, everyone. This is Amy. I'm a part of the tech team. I just want to give everyone a heads up about how the next sessions are going to work, because they're going to be just a bit different. If you go back to the session gallery, you'll see 3 different sessions to choose from. All 3 sessions are going to be repeated every 30 minutes. So basically, you can join into any 3 at 1, 1:30, or 2 o'clock Mountain Time. You can watch them in any order that you choose, but we'd love for you to watch all 3 because they're all very interesting topics. So thanks again, and thanks to our speakers, and we'll see you in the next sessions.